trial
This
is based on.
conference will now be recorded.
Okay.
On which clinical trial is based, and what type of people participated in the trial. Like it includes procedures, medication, dosages, and the length of study.
Who developed the protocol?
Okay.
Yeah.
2 case report form, CRFs.
Right. So protocol, you said protocol. So protocol, it's a plan to conduct the study, set of rules, set of procedures based on which your entire clinical trial will be done. Okay, it's a plan to conduct the studies. It will give you all these, let's say, so it will give you the design, methods, purpose, designs, methods, purpose, and
Hello?
hello. Hi, Ragichi.
Oh, hi, Chandra. Did you hear from Pre-Select?
Yeah. She
Okay.
messaged me. Yeah, thank you. So today we are discussing protocol, okay? So protocol is a plan, procedures, set of guidelines to conduct the clinical studies. In short, entire study will be explained in the protocol. Okay, but before we go to protocol, I would like to explain ICF, informed consent form. Informed consent form. So what is informed consent form? So this is a form by which information will be given to the subject and will take consent from the subject. ICF information will be given and will obtain consent, voluntary consent from the subject. Will obtain voluntary consent from the subject. That means explaining everything, risk, benefit, duration, study purpose and all will explain to the subject. Based on all these information, they will decide whether to participate or not to participate. If they are willing to participate, they sign the form. They sign the informed consent form. Okay. Let us see. You can see informed consent form. While preparing informed consent form, while getting the consent, we follow the 45 CFR Part 46. What is a CFR? You know CRF. CRF is a case report form. Case report form, which is used to collect the data. Which is used to collect the data. Okay. But what is CFR then? CFR is
Code Federal
Code.
Regulation.
Federal regulations. These are the regulations which are given by the US government. Federal Executive Department of US government. Not only into this clinical side. Okay, CFR guidelines that you have like for broad areas. CFR guidelines for the broad areas. Let's say Title 1, one topic. Title 2, one topic. Title 3, one topic. Title 4, one topic, like that. So Title 1, General Provisions. Title 2, Grants and Permissions. Title 3, President. 4, Accounts. 5, Banking, like that. So, entire CFR, we have total 50 titles, out of which 21 is a title which deals with food and drug. Total we have 50 titles. Out of all the titles, 21 is a title which deals with food and drug. That means anything related to food and drug which will be getting title 21. So title 45, public welfare. Title 45, public welfare. Hello. I'm audible now?
Yes,
It's a
ma'am. Yes. Yes.
right. So 45 deals with public welfare. In that part 46 will be with informed consent form. Okay. So remaining all titles we will not use. Most of the times we use 21 only, rarely 45 because 45 related to public welfare. Informed consent also somewhat related to public welfare. That's the reason it is explained there. Okay. Informed consent is a process by which participants voluntarily confirms his willingness to participate in the clinical trial. So informed consent describes rights of the study participants, including details about the study, like nothing but we need to explain all the details about the study to the subject. Participant then will decide whether to sign or not to sign the form. After going through all this information, they will decide whether to sign or not to sign the form. Informed consent form to be dated and signed by both the parties. In case if participant decide to join. Informed consent form must be in local language. It is a continuous process. It is divided into 3 copies. So first copy will be given to subject. Second copy along with the subject medical records. Third copy, original copy. Third copy, original copy will be for the investigator. So I have 2 parts here in the informed consent form. One is information sheet, other one is consent sheet. Part 1, information sheet. Part 2, consent sheet. Part 1 will explain all the details about the study. Part 1 will explain. So we have to provide written information to the subject. What we are going to perform, what we are going to do. So how many number of total patients involved, benefits, risk, any alternate treatment available. So these are all explained in the part 1. So here in the part 1, we include a statement. That means we should tell them this is the experimental study. It's not observational. It's a clinical study. We are conducting the research. So the statement that the study involves research. And how many days? They explain the purpose of the research. How many days patient will be in the study? How many days patients will be in the study? And what are all the procedures to follow? What are all the tests to be done? And risk, benefits, alternate treatment available. In case for the subject, any alternate treatment available. It's alternate treatment, risk and benefit, compensations, and treatment available to the subject in the event of trial-related injury. In case of trial-related injury, compensation treatment available to the subject. And statement that the participant, like subject participation is completely voluntary participation. Will include one statement that the subject participation is voluntary participation. Okay, so we'll include a statement that stating subject participation is voluntary participation, and we include even the statement about confidentiality of the records. Person to contact for further information, or in case if they want to know, like what to inform trial related injuries, whom to contact. So whom contact person name, person to contact for further information. Okay. So yeah. So that is part 1. Part 2, consent form. Consent form must be presented to the participant and signed before the subject can take part into the study. Okay. So if the subject is educated, name of the subject, signature of the subject, date. Date here it is given DDMMYY, but it's not like that. Any defined format you can mention date. So signature, subject signature is important along with the date, and investigator signature also important along with the date. Second case, if the subject is illiterate person. Okay. If the subject is illiterate person, we should have witness, name of the witness, signature of the witness, and name of the subject, thumb impression of the subject. Okay. In third case, if the subject is child. If the subject is child, name of the parent or guardian, signature of the parent or guardian, date. Investigator name, signature, date. Next we have protocol. So this is what informed consent form. Informed consent form will explain the study. Based on that, we'll decide whether to participate or not to participate. So based on that, they will sign the form. Next, protocol. So protocol is a document, describes objective, design, methodology, statistical consideration, organization of a trial. Okay, the complete details of the study. The study should be explained in the detailed protocol. So clinical trials are based on set of rules called protocol. Protocol will describe inclusion, exclusion. Means what type of people to be included, what type of people to be excluded. In overall, what type of people that you recruit. That will be given in the protocol. And schedule of tests, procedures, medications, dose, length of the study, how many days that you're conducting study, and how many days the patient will be in the study. General information, protocol title, protocol ID, and approval date. The question is, who will approve the protocol? questions. Who will approve the protocol? Who will develop the protocol?
By sponsor?
So developed by sponsor. Who approve the protocol then? IRB.
IRB. Yes.
Right. Subhashini, you don't have class today. 10:30 you have, not now.
Okay, ma'am.
So develop, yeah. So approved by the IRB. IRB, IEC. Independent Ethics Committee, Institutional Review Board. Both are same, but IRB that we use the term in US, IEC, Europe countries and other countries. Europe and other countries. Clear it? So approve the protocol, approved by the IRB, and protocol will be developed by the sponsor. The date will be there, approval date. Protocol title, protocol ID, protocol approval date. So name, name and address of the sponsor. Okay, name, address of the sponsor. And monitor. Monitor is a CRA, Clinical Research Associate. He is also called monitor. So name, title of the person who is authorized to sign the protocol, protocol amendments, and name, title, address, telephone number of the sponsor's medical expert. Name, title of the investigator responsible for the trial, and address, telephone number of the trial site. Address, telephone number of the trial site. Okay, so every sponsor will have expert, medical expert, to look at the activities. So name, title of the person authorized to sign the protocol, amendments, name, title, address, telephone number of the sponsor's medical expert. Okay, so name, title of the investigator, and address, telephone number of all the participating sites, clinical sites. It's all about like address, contact details of the teams and all. And other departments, clinical labs, central labs, local labs, other departments like medical, technical department, institutions involved in this. So summary of findings from non-clinical. So which potentially have clinical significance. And summary of known potential risk, benefit if any, to the human subjects. The background information means whatever the information that we get from the preclinical studies that will have potentially have significance, and we also even get background details from the previously conducted clinical studies. Let's say now we are conducting now we started clinical trial phase 1. We will have background information from preclinical. If you are conducting phase 2, we'll have background details from preclinical and phase 1. If you're conducting phase 3, you will get the background information from phase 2, 1 and preclinical. Okay. So this is what? Background information. So background information um, will even include the summary of known potential risk. Okay. So potential risk, benefits, if any, to human subjects, justification, route of administration, dose, regimen. Okay, so these are all treatment periods, and the statement that this trial will be conducted in compliance with the protocol, in compliance with the applicable regulatory requirements, other requirements, and what kind of population to be studied, description of population to be studied, references to literature, and data that is relevant to the trial. In overall, which will provide the background. In overall, which will provide the background. That means when you start conducting clinical study, we must have background. That background that we get from clinical, preclinical, clinical, preclinical literature. Okay. And so we'll have trial objectives. So what are the objectives and what is the purpose? You need to know definitely objective. You need to know the purpose of the study. You can easily understand by the name purpose of the study. Clear, right? The purpose of the study, why we are conducting, and we need to understand the objectives like primary and secondary objectives. So generally, can you tell me what is the main objective of the clinical studies?
Pharmacology and toxicology.
Okay. Anything else? Pharmacology, toxicology?
To evaluate the safety parameters.
Right. Safety and
Efficacy.
safety as well, efficacy of the drug. Right. So let's say primary objective is safety. Let's say primary objective is safety. So primary objective safety, secondary objective efficacy. The question is, how can we reach this one? How can we reach this? Any idea? How can we find the safety? That means what parameters that you require to assess that the drug is safe?
Adverse effects.
Right. Adverse effects. Based on the safety data, based on the list of adverse effects, based on all the data whichever collected about side effects, adverse effects. So based on the objectives, we have endpoints. We have endpoints. So what are the endpoints? These are the parameters, questions, data, which is used to assess the primary objective. So we'll have primary endpoint, secondary endpoints. So if you have two objectives, we'll have two endpoints. Primary endpoint is to assess primary objective. Secondary endpoint is to assess secondary objective. Okay. So let's say secondary endpoint efficacy. How can we reach efficacy? What kind of parameters that we require to assess that the drug is effective? So what we do here, it is based on the study, specific study. Efficacy will not be common, right? So let's say we are conducting diabetes, or let's say we are conducting the study on anemia. We are conducting study on anemia. So what is the secondary endpoint then? Primary endpoint is safety. So primary objective is safety. Endpoints are adverse effects data, lab data, any safety data. But efficacy, let's say if you are conducting trials on anemia, what can be the efficacy endpoint? Means what can be the secondary endpoint? How do we know the drug is effective in case of the trial is on anemia? What kind of parameters that we require here?
Hemoglobin levels.
Right, we'll mainly consider hemoglobin levels, HB percentages. And other factors also we'll consider along with HB percentage. Clear, right? Primary and secondary endpoints. So we'll have objectives. Based on objectives, we'll have endpoints. Endpoints are the parameters. Parameters, analysis parameters, variables, which are used to collect the, which are used for the objectives analysis. So these are analysis endpoints. How to analyze safety and efficacy means primary objective, secondary objective. So specifying all these objectives and description of the design. Okay. And design, type of the study, design of the study. So which type of the study to be done and which design to be followed, whether it is single, double, triple, open, and whether it is placebo control, active control, whether it is parallel, crossover, factorial. Okay, we'll have all these. And we'll have inclusion, okay, description of all the trial treatments, and as you can see here, packaging information, labeling information, dose form. Okay, so these are all details that will be included. Expected duration of the participation, how many days subject will be in the study, and description of stopping rules in case if you have any discontinuation criteria, accountability procedures, and maintenance of the trial, codes, randomization codes. If you're going for randomization, each and every drug will be identified with some code. How to maintain these codes and how to break the code. Okay, stopping rules, discontinuation criteria, accountability procedures for the products, maintaining randomization codes and all. So explaining inclusion, exclusion, withdrawal criteria, when to withdraw, how to withdraw, what type of data, timing of data, whether and how the subject ought to be replaced, and follow up for the subjects after withdrawal. So treatment of the subjects. So the treatment products, dose, in duration, treatment period, including follow-up, all these. And what are all the medications which are permitted? What are all the medications which are not permitted? Before the trial, during the trial.
Excuse me, ma'am.
Yes.
Ma'am,
Santoshi, you can chat. You can send me chat box. Yeah.
actually, I'm sorry, ma'am. I didn't check my WhatsApp, ma'am. So can I join? Shall I join at the 10:30 class, ma'am?
Yeah.
I'm logged
You,
off now.
no problem. You
Sorry.
can log. Yeah, no problem. No problem. Yeah. Right. So these are all the treatments of the subjects. Next, we have assessment of efficacy, assessment of safety. So we'll have list of all the efficacy parameters here. Which parameters that you're going to use in the analysis of the safety, analysis of the efficacy. Both lists will be there. After adverse effect, each and every patient will be followed up to know the outcome of the adverse effect. So what type of follow-up, duration of the follow-up after adverse effects. Let's see, statistical considerations. Criteria for termination, level of significance to be used while analyzing data. Number of participants, how many number of subjects plan to be enrolled. and description of the statistical methods employed. Interim analysis, the timing of the interim analysis, in the middle of the trial if you want to go for analysis, when to go, what is the criteria for it, any procedure for missing, unused, spurious data, and procedures for reporting any deviation from the original plan. Selection of subjects to be included in the analysis, all randomized, all dosed, all eligible subjects. So quality control, review, quality control, quality assurance. These are all the protocol contents. Let us list protocol contents, because I'm going to explain all these things with the example. So mainly protocol will explain title page. In the title page, mainly title of the protocol and contact details of all the persons, respective persons. So whichever important persons. Most of the times we'll have contact details and the title of the study, approval date, versions if any. Second one will have background information. Background information is important because you need to understand the study based on the previous studies. Okay, so this is background information. Third one, trial objectives, purpose. Objective and the purpose of the research. This is important. Fourth one that we have, treatments. What was the fourth one? Fifth one is inclusion, exclusion. Fourth one, explaining designs and all, right? Yeah, trial design. Explain the trial design, how we follow the trial. Explain the trial design. Fifth one, eligibility criteria, inclusion, exclusion, and withdrawal criteria also they will give. Inclusion, exclusion, exclusion, and withdrawal criteria. Sixth one, treatment of the subjects. What are all the treatments available for the subject? Those are all will be listed here. Treatment of subjects. And we have assessment of safety, assessment of efficacy. Safety, efficacy. Okay, so assessment of safety, efficacy, and statistical considerations, as well quality control, quality assurance. Okay, so this is what your like contents of the protocol. I'm going to show you one example now. Okay, I'm going to show you one example now. So with whichever the protocol that you see, first you need to understand the background. So what does it mean? Whichever the protocol that you want to understand, whichever the list of contents that you need to know. First of all, that you need to know the study details, background of the study. Why are we conducting the study? Okay, the background of the study that you need to know. That means what is the disease? Why we are applying this clinical trial to that. Okay, clinical trial, why we are implementing there. You need to know all these details first. Okay. So whatever the contents that we discussed till now, I'm simply going to explain. Simply going to explain. Example protocol. Let's see. I'll take one example. I'm going to explain protocol in that example. Okay, so this is another study that I'm not going to open this one. So this is an easy study. You can see here. So this is protocol title, if you can see. First, let us understand the title. Okay. So in the title page, what we have? Address, telephone number of all the concerned departments, concerned persons, title of the study, and approval information. As you can see, by the name, investigation, evaluating efficacy of full thickness placental allograft. Clinical investigation evaluating efficacy of full thickness placental allograft. So what is the objective here? By seeing the title, can you tell me objective, Nihal?
I actually don't.
Objective of the study? No?
Revita? Revita?
Revita is the product name.
Oh, okay.
So I'm asking objective of the study. That means why we are conducting this study. Objective means to evaluate the efficacy. So why are we conducting the study? Evaluate the efficacy. Efficacy is the end point. Okay, efficacy is the primary objective. So what we are doing here, we are evaluating efficacy of full thickness placental allograft. So objective is efficacy. We are going to evaluate the efficacy of this product. What is the product?
Revita?
Revita. Product name is Revita, but the product is allograft. Let's say I'm taking drug Ramistor. Ramistor is a registered name, but it's a drug. Similarly, Revita is a registered name. This is the product. Full thickness placental allograft is a product.
Sorry to cut you off, but so in the market, this drug will be known as Revita or Allograft?
Let's say when you say drug, you will have a lot of drugs, right?
Yes. Yes.
That is, a drug is a common term. When I say some drug name, paracetamol, that is a registered name.
Okay.
Like
So
drugs.
is that a paracetamol? Is it registered to registered from the company, or is it registered from the FDA? I mean, to the FDA.
Anything, trade name. Let's say metformin. You take the drug metformin. We have so many trade names for that. Glucophage is one of the trade name. Glucophage is one of the trade name. But what is a glucophage? Is it a drug? Is it a product? Is it a device? It's a drug.
It's a drug, right?
Drug is a common term.
Yeah, because I take metformin, so I know about that.
That's what that I'm explaining. You can see here.
Uh-huh.
Drug is a common term. Here also placental allograft is a common term. That means we have many placental allografts. The registered name Revita. Like glucophage registered name, but it's a drug. Revita is a registered name with a STIM lab. Okay, allograft is a common term. We have so many types of allografts with so many names. But our product is Revita. Clear, right? The difference between allograft and Revita?
Yes.
So placental allograft, we have so many. But Revita is a registered name with the STIM lab. Now you might have a question. So I am saying Revita is already registered name, means already in the market. So if it is already in the market, if it is already released into market, so Revita is a already marketed product. Then my question is, why are we conducting the research? Why are we conducting clinical study for this? It's already marketed, already available in the market. Then why are we conducting the clinical trial on Revita?
Is it because of the side effects? I mean, they want to study the side effects with all on the patients?
No.
Because during the clinical trial, it was conducted for a certain number of people or a certain age or number of people. So, but now that it's in the market, it's for everybody. So they want to know how the drug is doing.
The comparison of standard drug.
Mm-hmm.
The
Not
comparison.
exactly. Not exactly. Okay. So let's see here. Okay, we are comparing, but for the different application that we are conducting here. Revita, already known for its use in the skin ulcers. Revita already in the market for skin ulcers, wound covering, skin cuts, deep cuts. So what is this placental allograft? Let us understand the product. So what is placental allograft? So placental allograft is a tissue-based product. It's nothing but tissue, placental tissue. Okay, so this is derived from the human placental connective tissue. It will look like this. It's not the product, it's not the device, it's not the vaccine. It's just a product which will be available like this in the sachets. And it will look like this when you open. So it's not the drug, it's not the product, it's not the surgical procedure, it's not the test. This is a allograft, is a 2 into 2 centimeters. Allograft is 2 into 2 centimeters, human like tissue based product. Human donor cell. It is also called tissue based product. This patch. Okay. So actually this patch is used for skin cuts. Wound covering. Let's say when you have deep cut on your skin, on your hand, usually we go for stitches. Usually we go for stitches. But this Revita will help you to avoid stitches. Without stitches, if you just patch this over wound, that will be healed off. So this is what tissue-based product. Okay. You understand the product right now? So we are not experimenting on drug. We are experimenting on allograft. That too, the product which is already in the market. Then why are we conducting the studies here? So for the new application, new purpose. The application is like if the like product already known for the skin ulcers, wound covering. But for first time, we are using in the surgical process. That's the reason we are conducting research. Okay, for skin ulcers, it's already in the market, already released. But till now, we didn't use the same product, Revita, in surgical place while performing surgery. Let us understand why allograft is important, why allograft is required, why allograft is important, why allograft is required. To elevate the, like why it is required in the surgical process. Let us understand the surgery. So till now what we did, we learned about the product. Because a product can be drug, it can be device, it can be new surgical process, or it can be new product. Here is the new product. So now we know about product, whether it is a new, already registered. If it is already registered, why, like what purpose it is registered? What purpose it is registered? So we know all of them. Okay. Now let us understand the disease or a problem. Why we are using this product? So this product is used in surgery. Now the experimentation in surgeries. Then why this surgery? What happens in this surgery? Let us understand. Okay. So surgery in the lumbar place. In the lumbar place. You see, in your spinal cord, mainly we have three parts: thoracic, cervical, and lumbar. Cervical is neck part, thoracic is middle part, lumbar is lower part. So in the disc, in the spinal cord, okay, so we have disc materials which will act as suspension to move. It will help you to move, twist. Okay. So that is called, that material is called disc. So disc bulge. You see, this is disc. This is normal disc. This is how your spinal cord look. Normal disc, degenerative disc, bulging disc. So in case of bulging disc, doctor will not suggest you to go to surgery. It will not interfere activities much. But when the same bulge is herniated. Herniated disc will interrupt your activities. What happens when the disc herniate? It will compress the spinal nerves like this. There is a nerve you can see. It will compress this nerve. This nerve, compressed nerve, it get irritated and cause severe back pain and leg pain. Not only back pain, lower back pain, even leg pain also there. You can see the complete portion will be affected, red color portion. Okay. So in the traditional surgeries before Revita, what they have used, which procedure that they follow previously before Revita. Now we are conducting the study for Revita, but before Revita, what was a normal procedure? Normal procedure, they'll undergo surgery. They'll undergo surgery. During surgical process, they will dissect this herniated portion. Means they will cut this portion. Herniated portion is there, right? They will cut the portion and remove it. The herniated portion will be removed. Now, if you remove and if you cut, there will be two open ends, right? If you cut that portion, there will be two open ends. The two open ends will be patched up with the Revita. Traditionally, you may go for surgeries, like you may go for stitching. In place of that, like when you have two open ends, we go for easily stitches. But because of stitches, there are the drawbacks. Scar tissue formation. Okay, so it will it's a scar tissue formation, and it leads to a reherniation also. So before we start this study, the traditional surgeries, what they do, they used to dissect this portion and they stitch these two open ends. But because of stitch, there may be scar tissue formation and the reherniation rate. They will be again started herniating portion. Okay, most of the people complain on back pain and leg pain even after surgery also. The reason is the scar tissue formation, and the people again started showing reherniated disc. Okay, so with the Revita, what they do, there will be two openings when you dissect this. The two openings will be patched up. Two openings will be patched up with the 2 into 2 centimeters Revita. Okay, that's the reason we are conducting research. Okay, so you need to understand the background of the disease. Whichever the protocol that you see, you need to check background of the information, disease information, and why we are using this, purpose of the research and all. Clear, right? Any protocol, not only this protocol. So this is a protocol we are taking as an example. So no need to know all these terminology also, like placental allograft, or let's say this terminology, disc related, lumbar related. No need to know all these terminology, but Whenever you see protocol, you need to go through all these details. Every protocol will have background details. Every protocol will have title. So now you understand the title, right? Efficacy of full thickness placental allograft. So what is placental allograft? It's a tissue-based product. Revita is registered name. Okay. And in lumbar microdiscectomy, outcomes. Lumbar means lower back. Microdiscectomy means surgery. Why we are going for surgery? When there is a herniated disc, bulged disc. In the surgery, what they do? They will remove that herniated portion. While removing, you may face two problems in the traditional surgeries. There may be scar tissue formation if you go for stitches, or there may be reherniation rate. The main objective here is the efficacy of the drug, efficacy of the product. Clear? Nihal?
Yes.
Geet. Is the title clear? Nandini Ogechi. Is it clear? The title.
Yes. Yes, ma'am. Yes.
Yes, it is.
So the title page will cover all these. Title page will cover title of the protocol, like investigator details and the sponsor details. Investigators explain, investigator name, like address, telephone number, fax, email, and all. Sponsor also will collect the same details. You can see the summary. So if you go to summary, you will understand almost half of the protocol. If you simply go through the synopsis of the protocol, you'll be able to understand 50 to 60% of the study protocol. In the summary, so we have title. In the summary, we have title, methodology, randomized, controlled, blind study. Randomized, controlled, blind study. Okay, so we are saying randomized. We are saying controlled. We are also saying blinding. But we don't know whether it is single, triple, double blinded, whether it is placebo control, active control. It is stating randomized, that's all. It is not given which type of control that we use, and blind study, whether it's single, double, triple, open. Okay, and duration 2.5 years. Duration will be mentioned, and site places will be mentioned. Where do we conduct these studies? See, most of the clinical trials are multi-center. List all the sites here. Primary and secondary objectives. Primary and secondary objectives. So let us see the objectives. You know what is objective. Objective is for what purpose that we are conducting this study. the main objective, main goal, main aim of the trial. So two aims here: to achieve, like to evaluate the efficacy, and to evaluate the efficacy. So, and to evaluate the reherniation rate in people who treated with the Revita. So reherniation means repeat of same herniated disc. When you dissect the portion, herniated portion, the disc again started a reherniation. Okay, number of subjects 182 randomized patients in 2 arms, treatment, control. Number of subjects total 182 into 2 groups, treatment group, control group. Okay, now inclusion, exclusion. So you know inclusion. So inclusion criteria, exclusion criteria, completely study based. Here the main inclusion criteria will recruit if the people suffering from back pain for last 6 months with a clinical diagnosis of lumbar protruding disc. That means whoever have the disc. So whoever are showing the bulging disc, like herniated disc. Exclusion also explained here. Inclusion, exclusion. That means which type of people not to include, which type of people to include. Let's say primary, secondary endpoints based on the primary and secondary objectives. Based on primary, secondary objectives. Okay, so this is synopsis. Now you see, understand the primary endpoint. Primary objective is efficacy. How can we know the efficacy here? How can we reach the efficacy? What kind of parameters you require to know the efficacy of the product, efficacy of the Revita? After surgery, they have to collect pain-related scores, pain-related questions. While bending, what is the pain severity? While walking, what is the pain severity? While sleeping, while bending, while kneeling. Okay, so we'll collect all these while working. We'll mainly collect the pain score while doing daily activities. If you have the pain score after giving the drug, before giving the drug, we can easily assess the efficacy of the drug, efficacy of the product. Here endpoints are the parameters. You can see back pain, leg pain measured by ODI. ODI is a questionnaire. We'll have a set of 10 questions in the ODI. And SF12 will have set of 12 questions in this form. So every time we need to give these two forms to subject and ask them to fill. Because in these two forms you have efficacy related questions. Okay. Second endpoint is reherniation rate. So reherniation means, what is reherniation? After surgery, people generally start again herniated disc. The same thing happen again, disc bulges and to form herniated disc. So that is reherniations. After surgery, after that to traditional surgery, without Revita, reherniation is a common problem. But here we are using Revita as a barrier membrane, thick placental allograft. So there will be less chance, we are expecting there will be less chance of reherniation rate. There it leads to low back pain, like lesser back pain, and there is an improvement in the back and leg pains. This is all the synopsis. It's a short summary about study. So these are all the detail. You can see the purpose of the study. Primary objective is to evaluate okay. Primary objective is to evaluate the efficacy of Revita, full thickness placental allograft, in improving low back pain and leg pain. So we are using Revita in improving low back pain and leg pain in the surgeries, microdiscectomies. Background. Microdiscectomy is a routine process to elevate herniated vertebral disc material, which is compressing. You can see if you understand this statements, you'll be able to understand the background. Why this process? Why this clinical studies? You will understand if you go through this one. So microdiscectomy is a routine process to elevate herniated vertebral disc material, nothing but disc, that is compressing spine and spinal nerves. So we are going for the surgery to elevate the disc, which is irritating, which is compressing the spinal nerves. And we'll do the surgery. But the two common problems from the surgeries: scar tissue formation, reherniation rate. These two are the common problems. Reherniation, scar tissue formation can lead to again symptoms even after surgery. Also, patient might be suffering from back pain and leg pain because of these two problems: reherniation, formation of scar tissue. But Revita is designed to prevent scar tissue formation because we are not going for the stitches there. It's a patch. No chance of scar tissue formation. So which will be expected to reduce the symptoms frequently seen in the surgery, traditional surgery. So this is background. Goals of the study. Okay. So goals, efficacy, improve the pain scores after surgery, and reherniation. Duration of study, 2.5 years. Product is described here. No need to go with the product information because every product, a different descriptions will be there. No need to go with the product description. So Revita is donated allograft placental tissue. It's an opaque white to off-white dehydrated material. It is a donated human tissue regulated by the US FDA as a human cell. These are all product details only. Product intended use. The Revita is used as wound covering, barrier membrane, over chronic acute wounds, including dermal ulcers, defects, including dermal ulcers and defects. See, product acquisition, it's not available at free cost. It is at free cost to the subject, but not to the site because it's not a clinical trial. It's not. It is clinical trial, but it's not the clinical trial for the new product. It's already existing product, but we are using for the new application, new purpose. That's the reason Revita allograft will be purchased by the Northside Hospital from TimLab at no cost to the patient. At no cost to patient, and Revita allograft will be purchased by Northside Hospital. Okay, so these are all the benefits and risk of the product will be explained here. No need to even go with this one. Okay. Let us understand the design. Let us understand the design of the study. 182 people. See the last statement. So design, we have 4 designs. Randomized and randomized as one design. Placebo control, active control, one design. Single, double or triple or open, one design. Placebo, like parallel factorial crossover, one design. So the study designs are explained here. You can see. So just go through the last statement and tell me which type of blinding it is. Is it single or double or triple or open? Which type of blinding it is? By reading the last statement, you will get to know.
It's a single blinded.
Single blinded. Nihal, why it is single blinded? Jeet. If your mic is working, you can tell me, Jeet or Nihal. Why it is single blind? Are you there, Nihal? Jeet? Ogechi, you can tell me. No, I can't hear you, Nihal. Maybe your face. Why
Because
is
it
it?
says the patients will not know that they will be receiving the Revita. So it only
Correct,
mentions the patients.
correct. Okay, right. So patient in the study will not know that they will be receiving the Revita. It is single blinded. Okay, and you see parallelly we are performing two surgeries. So here we are recruiting people, 182 people. These 182 people randomized into two groups. One is treatment group, 91 patients. Another is control group, another 91 patients. So treatment group of 91 will receive Revita allograft. Okay, these people will be treated with the Revita allograft. Then other group of people, 91 people, undergo surgery without any additional application, without Revita. So total 182. We have two groups. One group receives Revita, another group receives no application. We are comparing nothing but we will know the better efficacy of the product if you compare with the control group. Control group means who receives nothing, no drug, no product. Whatever the traditional surgery that we are following, we follow the same procedure for 91 patients. But another 91 patients will be treated with the Revita. Okay, so this is study design completely explained here. What about data collection? What about data collection? So here the data will be collected on OBRD systems. This is data collection tool. So they use, like, depends upon the study. They may use different collection methods. Sometimes they may completely depends upon electronics. Sometimes they may completely use paper forms. Mixed. Here mixed. As a primary source, they're using electronic capture. As a secondary source, they're using paper. This is data collection tool. In that, the physicians will collect the ortho-related data. As you know. Okay, so inclusion, exclusion, you know, inclusion, exclusion explained here. What about recruitment methods? How the people recruited? Subjects will be identified through the existing patients of Polaris Spine and Neurosurgery centers. Primary investigator, co-investigators will be performing the surgery for both control and treatment groups. The study, all the studies generally will be listed in the clinicaltrial.gov. Data collection, reporting. The data will be immediately collected before the surgery after the surgery. Before surgery, it is called baseline data. They have to collect the data before surgery. After surgery, at first month, second month, third month, 6th month, 12th month, 24th month, they must collect the data. Okay, so this is data collection. How they are going to collect the data? They collect the data by like subject visits, physical visits. They may collect the data via phone, email, phone surveys. So here we use two forms, ODI, SF12. ODI is a 10-topic questionnaire. SF12 is a 12-topic questionnaires. Okay. Any changes? 12 like SF12 will be any changes in ability to perform the daily task. ODI, disability index, quantify the disability due to low back pain. Okay, so this is what data collection. Data will be collected in all these points before the surgery and after surgery also. For 2 years, the follow-up data is collected. So what is the expected outcome from the study? Sponsor expectation that short-term, long-term evaluations of application of Revita will show improved patient outcome. Improvements in both ODI and SF12 are expected from the treatment group compared to control group. Interpretation, analysis will be described in later further pages of the protocol. Mainly, we're expecting that the improvement in low back pain and leg pain or ODI SF12 scoring from the treatment group compared to control group. Treatment group means whoever treated with the Revita. Control means whoever treated with traditional surgery without any application, without any tissue application. Adverse reactions. So there is no expectation of any adverse outcomes or reactions due to patient being treated with Revita, because the surgery is not the new surgery, new surgical process, right? The surgery is already known method. And Revita is also already released a drug already in the market. So here the Revita allografts are minimally manipulated. No additives, no sedatives, no chemicals that would be expected to cause the adverse effects. So we are not expecting any adverse effects. And two, on well-known methods, like one, product is already used in the market, and the surgery we know already known procedure. Reasons for the withdrawals. So as it is voluntary participation, subject can withdraw from the study at any time. At any time. Okay. So this is reasons for the withdrawals. Subject can withdraw, may be discontinued from any time point. Okay. In case of the investigator or subject sponsor feels that it is not subject best interest to continue. You can see the possible reasons for the discontinuation. Screen failure, withdrawal of consent, not compliant with the procedure, adverse events, protocol violations, loss to follow, sponsor request for the early termination, death. So all subjects are free to withdraw from the participation at any time, any reason, specified, unspecified. But patient should be followed up for the withdrawal reason. We need to know the reason. For the reason, we need to follow up the subject. In case if the subject is withdrawn from the study because of the adverse effect. If the subject withdrawn from the treatment due to adverse effect, subject will be followed, treated by the investigator until that abnormal parameter has been resolved. Means until they recover from the adverse effect. So investigator must make every effort to contact the subjects who lost to follow. What about withdrawals? In this study, the withdrawals, patient who withdraw not to be replaced. See? Subjects who prematurely discontinue from the study. These people are not to be replaced in the study. In this study, it depends upon the protocol. Other studies, they may accept the they may replace the subjects, withdrawn patients. But here, withdrawn patients will not be replaced because it is surgery. Whenever the patient withdrawn, they cannot go on conducting the surgery for new people, right? It's difficult. So, not to be replaced. Withdrawn patients, not to be replaced. In case if the subject, the sponsor, in case if they want to terminate the study, complete study if they want to terminate. When can they terminate? If they terminate, they have to inform the respective bodies, regulatory authorities, IRBs and all. So when can they terminate the study? It will be given in protocol. See possible circumstances where the discontinuation or termination suspension include. You can see unexpected, significant, unacceptable risk, demonstration of efficacy, insufficient compliance, and data that is not sufficiently complete, determination of futility. Study may resume once the concerns about safety, protocol compliance are addressed. It may be restarted. So the ones they discontinue the study, suspend the study. Once all the issues are resolved, they may continue the study, resume the study. These are our methods and schedules. The first point is eligibility criteria. So we need to call the patients, ask them to give their details, and decide whether patient is fit to participate in the trial, good to participate in trial. This will happen by considering inclusion-exclusion. After that, we'll inform the subject about the study. We'll take the consent. Once consented, then we'll follow randomization process. Randomization means randomly assigning drugs to the randomly assigning subjects to the groups. Means we don't know which patient is assigned to which group. Then after that, treatment. The treatment group receive Revita, control group receive no applications, traditional surgery, standard surgery. Baseline visit will happen before the treatment. Before the treatment, baseline will happen, that to 3 months before the treatment. See, it is showing -91, 3 months before it is started, baseline visit, and ended before 1 day, 91 to 1, -1. Day 0 is a treatment day. On the day the surgery was happening. So during the baseline, they have to collect all these details. See. During the baseline, they will collect all these details from the subject. On treatment day 0, they will perform the surgery. Okay, randomization of qualified subjects will be into OBRD system. OBRD system is a data collection system, data collection tool. So 91 member control group, you can see 91 control group will undergo typical surgery, typical microdiscectomy surgery, and treatment group 91 with the addition of Revita application. So treatment visit, treatment will happen on day 0. So follow-up visits, we have all these. After discharge, follow-up will be done at following time points: 1, 2, 3, 6, 12, 24. And during all the follow-up periods, patient will be given 14 days time period. Patients will have grace period of 14 days after each time point, okay, to complete the follow-up. Assess for the adverse events, complications, and head documenting medications, perform the physical examination, ODI, SF12, evaluate for the reherniation. Those are all during follow-up. Final study visit, last day of the visit. We again collect this data. In case of unscheduled visit, means unscheduled, when will this happen? In case of adverse effects. So, but any point, any time point other than mentioned above, the patient can visit the site and inform their problem. In case of early termination, in the middle of the study, if they want to withdraw from the study. During early termination also, we have to collect all this data. Okay, randomization. The subject who meet all inclusion-exclusion criteria will be randomized on treatment day into 1:1 ratio to either the treatment group or control group. So generally, this randomization plan will be given by biostatistician, implemented by the site person into the OBRD system. Okay. And statistical analysis plan is explained here. Endpoints, primary, secondary endpoints, and what type of people that we include in the analysis. Whether we are going to analyze all the dosed people, all randomized people, all evaluable people, like that. Assessment of safety. Your assessment of safety is not a main concern, right? Assessment of efficacy is a main objective. Safety is also given here Okay, as we are not expecting any side effects here. Okay, but any adverse effect they can inform, they can report. So when do we collect these adverse effects? Adverse effects are collected here during the study, right from they assign the consent form until 7 days for non-serious, until 30 days for serious. Even after completion of the study also, patient will be monitored for 30 days, 7 days. For adverse effect, detailed explanation will be obtained from the subject. So you know what is adverse effect: unwanted, unintended, untoward medical occurrence. So you know when can we call it as serious, right? When it is fatal, life-threatening, hospitalization, permanent disability. Definition of all the serious adverse events and definition of unanticipated event, unexpected event. So whenever you have new event, new adverse event, we need to know whether it is expected to cause, expected to occur, or is it a new one. So whether it is anticipated by nature, whether it is already known by nature, by frequency, by intensity. If it is already known, it is called anticipated, otherwise unanticipated. The severity, mild, moderate, severe, and relatedness with the study product means a relationship between drug and the event, whether drug caused event or any other reason. Okay, definitely related. Means there is definite relationship between drug and event. Probably, maybe, may not be, possibly, maybe, may not be. Unrelated. Definitely not related. The adverse effect is not related to the study drug, study treatment. You know, expectedness, whether the adverse effect is expected or unexpected. It is considered unexpected in nature, severity, frequency, if it is not consistent with the risk information, whatever the information that you have, whatever the information that is previously reported. Based on the previous data, we'll get to know whether it is expected or unexpected. What are the time periods? Principal investigator will record all the reportable events with the start dates occurring any time after informed consent is obtained until 7 days or 30 days period after last day of the participation also. So whenever the serious adverse effect occur, investigator must notify the sponsor within 1 working day. Okay. So whenever the serious adverse effect occur, within 24 hours time to sponsors, 7 days to FDA, 15 days for other serious. Fatal, life-threatening, 7 days. To IRB within 10 days. So whenever the serious adverse effect occur, 24 hours to sponsor, 7 days to FDA, 10 days to IRB. Pregnancy also will be reported. Safety monitoring boards not required. See, the formal data safety monitoring board will not be needed for the study because we are not expecting any adverse effects here. Data monitoring boards will be there. Data monitoring team will be there, appointed by the sponsor. They will visit the study. They will visit the site to check the study, how it is going on. Mainly, we should make sure the study should be conducted as per the protocol, as per the GCP, as per the regulatory requirements, concerned regulatory requirements. Okay, but sponsor will decide who will perform this monitoring, at what frequency, at what interval, at what level of detailed monitoring should be done, how to monitor, how to distribute the monitoring reports, how to exchange the monitoring reports. So mainly we should know the clinical trial data is valid. Mainly the clinical trial data is conducted according with all these. How to handle data, who will enter data, study staff will enter the data. Data entry will be completed into OBRD electronic system. Later it will be exported to SAS teams to analyze it. And IRB as a institutional review board. We'll discuss in detail later about this one. But as of now, all the documents, amendments to documents, changes of the documents will be approved by the IRB. Consent process explained here. How you reach the subject, what kind of information that you have to give to subject, how to take the consent. All these will be explained here. In case of any protocol deviation, non-compliance with the protocol, nothing but. If you're not following protocol, if subject is not following protocol, that should be here in the protocol deviation. Let's say so-and-so drug is prohibited drug, but still the subject is taking the drug. It is protocol deviation. It should be mentioned on the CRF. Laws and regulations as per the company, publications and data sharing also as per the company, and study personnel roles. Okay. Explained here. Principal investigator responsible for study related issues. Co-investigator screen, consent, follow. Almost same as study coordinator. Project manager look after the complete project, data management analysis activities. Statistician for analysis and data manager for management activities, data management activities. And you have schedule of events, you can see here. Okay, these are all schedule of events. So this is protocol. No need to know each and every term from the protocol, but you just need to understand the contents of the protocol. So what are all information that you get from the protocol? Okay, so you can compare these contents, whatever I have explained here, and compare with this example so that you can easily understand this protocol. So I'm going to provide another protocol so that you can look at that